Hi everyone, my name is Annie Huo, and this summer I worked with the Laird Lab to investigate the parallels of chick and mouse primordial germ cell migration. So, primordial germ cells, or PGCs, are precursors to mature germ cells which later produce gametes through meiosis. So, PGCs go through a rigorous migratory process early in development to reach their gonadal niche. This migration is conserved across many species, including mouse, humans, chick, and Drosophila. These model organisms have been used to decipher the mechanisms and molecular nuances of the PGC journey that helps them reach their final mature fate. Mice are widely used in reproductive research because of their evolutionary similarities to humans, but it's hard to investigate the process live in vivo. So this is why in the Laird Lab we are developing a xenotransplantation assay that allows us to observe migration in vivo. By transplanting mouse PGCs into the chick's unique vascular system, we can observe the live migration process and gather better information about the molecular nuances. Understanding why and how mouse PGCs can reach the chick's developing gonad can help us understand the migration process of human PGCs and allow the advancement of research for critical reproductive health issues. The setback to this transplantation method is that currently there's not enough tools to collect accurate data. So this is why this summer I worked to test different antibodies and staining protocols in hopes to achieve clear stainings. So this is a quick overview of the process for testing different antibodies and protocols. So here are chicks, and they were harvested at the stages from HH23 to 26. The AGM, which is this region right here, was then extracted and prepared for fixation. Similarly, in mice, embryos were harvested at E9.5, and then the GFP positive region was extracted under a fluorescent scope. So our mice have a GFP positive transgene, which acts as a control for mouse PGCs in the future. So after harvesting and dissecting, we fix them in either PFA, methanol DMSO, or glyoxal, and then they're then stained with the standard immunostaining whole mount protocol, cleared, and then imaged in a confocal microscope. So here are some of our samples. This is a chick AGM, which is staged at HH26 and fixed in PFA. So this is a 3D image with many slices stacked together to form a three-dimensional replication of her figure. And we use SOX2, which is an early nucleic pluripotent cell marker, and DASL, which is a germ cell remembering marker. And we tested these because we found that they were expressed at this time point in previous papers. We can see that they did not work because there are no clear definitive structures stained compared to the control SOC channel. And then here's another sample, which is also a chick AGM fixed in PFA. And this is laminin, which is a blood vessel marker, and we can see it's stained pretty clearly in a region that's known to have high blood vasculature. So this is a mouse AGM, and it's E9.5 fixed in PFA, and the SSEA, which is usually a reliable germ cell marker and used as a control, actually did not work in the sample, but we know that the germ cells are here because we confirmed the location of the GFP positive cells under an epifluorescent scope. And DAPPA5 and SDF were used. Both of them had some signaling, but if we look here, this is a 3D image. There's really high background and you can't see the structures very clearly, but when we look at the slice image right here, there are clear definitive staining structures. And then over here is SDF1, and we see some signaling right over here, but it's not as robust. And then this is just a video. So, as a summary, previously we used methanol DMSO as the primary fixation method, but this summer we tried a new fixation method, PFA. We tested many germ cell related antibodies, and then the ones that did work were in PFA were laminin, DPP5, GFP, and SDF1. If I were to continue to pursue this project past the summer, I would utilize this new panel of antibodies to analyze the migration pathway of transplanted mouse PGCs in a chick-vax literature, confirm the location of the mouse PGCs with dual markers, and do quantitative 3D analysis of samples using new antibodies. So I'd like to thank the Laird Lab for providing a really supportive environment this summer for me, and especially my mentor, Lena Fonza, for helping me through all these stainings, and also the Schneider Lab, which provided us eggs and other resources, and Lowell LSRP for providing this opportunity and letting me have this wonderful experience. Any questions? All right, thank you.